Okay, let's take a look at ethanol metabolism. Primary location for ethanol metabolism, of course, is in the liver and the hepatocytes. Ethanol is a very small and simple molecule, and being so, because of this size and the chemistry of it being slightly polar, it is easy to pass into the cytoplasm. It passes through membranes by simple diffusion, and enters the cytoplasm for the first reaction of ethanol oxidation. So the first reaction is to take the ethanol molecule and to oxidize at this carbon. And we are now generating an aldehyde. This is acetaldehyde. The enzyme that does this is alcohol dehydrogenase. And of course, being a dehydrogenase, it is mediating a redox reaction. So at this very first step of ethanol oxidation, we are generating energy in the form of NADH. Now the next step is to transport acetaldehyde into the matrix of the mitochondria. In this location, the acetaldehyde can undergo further oxidation with acetaldehyde dehydrogenase. Again, because this is a dehydrogenase enzyme, it is going to mediate a redox reaction, and we are generating a second NADH molecule. The resultant of this oxidation is going to be acetate. Now at this point, the acetate has two fates. The first is that the liver can use the acetate for energy if it needs. Second, it could be transported into the blood to extrahepatic tissues. In either case, the utilization of acetate is going to be mediated through a reaction with acetyl-CoA synthetase. This enzyme will take the acetate, it will add, of course, coenzyme A, and the energy to drive this reaction will be through ATP utilization. What we will end up with in the end is an acetyl-CoA molecule. The acetyl-CoA, of course, can proceed into the TCA cycle via condensation with oxaloacetate. That's pretty much it for ethanol metabolism. A couple of points to note: the acetaldehyde, the aldehyde group of acetaldehyde, is very reactive, and extensive alcohol consumption leads to reactions with these aldehyde, and eventually this can cause hepatotoxicity. Because the aldehydes are very reactive, they'll bind to anything indiscriminately, including proteins and lipids, and disturb their function, causing toxicity. A couple of genetic notes. Individuals that have been identified with acetaldehyde dehydrogenase mutations that decrease activity of the enzyme or eliminate its activity, these individuals will have an increase in their concentration of acetaldehyde. Because of this, these individuals have no reported cases of alcoholism, for the simple fact that acetaldehyde is such a toxic chemical that this is the chemical that causes our hangovers. The acetaldehyde reacts with the CTZ center in the brain and induces nausea and sometimes vomiting. Again, these are unusual chemicals that are detected, and the body wants to get rid of them. On the flip side, acetaldehyde dehydrogenase mutations that cause an increase in activity. These are called gain-of-function mutations. These individuals will, of course, have a lower concentration of acetaldehyde, and these individuals have a higher risk of alcoholism, simply because they are processing the chemical that causes the nausea and vomiting much faster and clearing it from their body. So they can handle a higher concentration of alcohol without the terrible side effects. Now, in the next video, we'll look at regulation of metabolism due to ethanol oxidation. This is much more complex because we will need to look at both the fed and fasting state in the liver and see what oxidation of ethanol is doing on these pathways.